Heteropolysaccharides are polysaccharides that contain more than one type of sugar units. So there are various types of heteropolysaccharides, and among them I have mentioned here four major types. So there's your glycosaminoglycans, where glycan refers to polysaccharide. In short, GAGs, which is also called mucopolysaccharide. This GAG or mucopolysaccharide is the most commonly found heteropolysaccharide. Second is agar, which is used for bacterial culture. Third is agarose, which is used during electrophoresis. And fourth is pectin, which is used as dietary fiber. Among these four types of heteropolysaccharide, the one that you need to know in detail from examination point of view is glycosaminoglycans or mucopolysaccharide. So let's deal with it. What I have here is GAG or glycosaminoglycan, which is also called mucopolysaccharide. The reason it is called mucopolysaccharide is because it is isolated from mucin, and mucin is the major component of mucus. That's why the part muco. Why? Because the polysaccharide is isolated from mucin. What's mucin? It is the major component of mucus. Now, heteropolysaccharide, they are made up of more than one type of sugar unit, right? So what are the sugar units found in GAG or mucopolysaccharide? So GAG or mucopolysaccharide is composed of repeating disaccharide units. Disaccharide unit. So what's this disaccharide unit which is repeating? Well, the disaccharide unit which is found repeating in case of mucopolysaccharide is made up of two sugar units, amino sugar and uronic acid. So the two sugar units, amino sugar and uronic acid, form one disaccharide unit which is found repeating in case of GAG. So let me draw it. Here's your amino sugar and then here's your uronic acid. Amino sugar and uronic acid together forms one disaccharide unit, attached to which is found another disaccharide unit which is composed of amino sugar and uronic acid, attached to which is found another disaccharide unit which is formed of amino sugar and uronic acid, and the chain goes on and on with repeating disaccharide units. Now this amino sugar which is present in the disaccharide unit can either be glucosamine or it can be galactosamine. Similarly, the uronic acid which is found in the disaccharide unit can either be glucuronic acid or it can be iduronic acid. And this is the basic composition of GAG. So what is this? This is the composition or the structure, basic structure of GAG or mucopolysaccharide. Now let's look at the properties of GAGs, because once you understand the various properties of GAGs, you will be able to automatically guess the places where GAGs can be found and their uses at those places. But first, let's look at its properties. So GAGs or glycosaminoglycans or mucopolysaccharides, they contain negatively charged groups such as carboxyl, COO-, sulfate, SO4-2-, and acetyl. Due to the presence of these negatively charged groups in GAGs, they are able to repel one another. So there's one molecule of GAG and here's another molecule of GAG. Both of them contain negative charge. The negative charge of one GAG repels the negative charge of another GAG. So they contain these properties of being able to repel each other due to the presence of negative charge. Another, GAGs, they can bind with large amount of water. And due to the presence of these two properties, property number one, due to the presence of negative charge, they repel each other. And property number two, being able to bind with large amount of water, the matrix or the ground that is formed by GAGs is gel-like. So they form gel-like matrix. And I hope you can guess the properties of a gel. What does a gel feel like? Gel has slippery property. It acts as a cushion. So it has cushion-like property. Due to slippery property of the gel-like matrix of GAG, it acts as a lubricating agent. And due to the cushion-like property of the gel-like matrix of GAG, it acts as a shock absorbent. How exactly do they act as shock absorbent? This is how. So GAGs, they are like sponge. If you squeeze a sponge, it decreases in volume, and if you let it go, it comes back to normal or it increases in volume. Similarly, GAGs, if they get dehydrated or water molecules somehow come out of its matrix, it gets squeezed. So here's your GAG, here's a ball of GAG, gel-like matrix of the GAG. If it gets dehydrated, it gets squeezed and decreases in volume. But once it gets rehydrated, or the molecules of the GAG are negatively charged, right? So they repel each other, right? So the molecules of the GAG that is present in the matrix, they repel each other, and as a result, it increases in volume. So due to this property of GAG being able to get squeezed and then expand, squeeze and expand, squeeze and expand, it acts as a shock absorber. And this is how GAG acts as number 1, lubricating agent, and number 2, shock absorber. From this, I hope you can guess where exactly are GAGs found and what's their function there. Now let's look at the various types of glycosaminoglycans, their composition, occurrence, and function. So there are 6 major types of GAGs that you need to remember, and they are hyaluronic acid, chondroitin acid, heparin, heparin sulfate, keratin sulfate, and dermatan sulfate. Again, there are 6 major types of GAGs that you need to remember, and they are hyaluronic acid, chondroitin acid, heparin, heparin sulfate, keratin sulfate, and dermatan sulfate. Now, we know that glycosaminoglycans, they are formed of repeating disaccharide units. And this disaccharide unit contains 2 different types of sugar, and they are amino sugar and uronic acid. Amino sugar is either glucosamine or galactosamine. Similarly, uronic acid is either glucuronic acid or ioduronic acid. Now, this is what you need to know. This is very, very important. The most common composition that is found in GAGs is glucosamine as amino sugar and glucuronic acid as uronic acid. However, there are exceptions to it. What are the exceptions? Okay, so all of them contain glucosamine as amino sugar except chondroitin acid. It contains galactosamine instead of glucosamine, and dermatan sulfate. It contains galactosamine instead of glucosamine. So C, D contains galactosamine instead of glucosamine. Another. So almost all of them contain glucuronic acid as uronic acid except dermatan sulfate. It contains ioduronic acid instead of glucuronic acid, and keratin sulfate. Keratin sulfate does not contain uronic acid. It contains galactose instead. And then there's your heparin, which contains both glucuronic acid and ioduronic acid. Let me repeat everything for you. So there's your amino sugar plus uronic acid making up the disaccharide unit. Amino sugar is either glucosamine or galactosamine, and uronic acid is either glucuronic acid or ioduronic acid. Almost all of them contain glucosamine as amino sugar except CD, that is chondroitin acid, and D for dermatan sulfate. Except chondroitin acid and dermatan sulfate, all of them contain glucosamine. Chondroitin acid and dermatan sulfate contains galactosamine. Similarly, almost all of them contain glucuronic acid as uronic acid except dermatan sulfate, which contains ioduronic acid, and keratin sulfate, which does not contain uronic acid. It contains galactose instead. Now let's look at the occurrence and functions of these various types of GAGs. So hyaluronic acid, it is found in synovial fluid and vitreous humor, where The function of hyaluronic acid is to act as lubricant, number one, so that there's no friction between the two so that there is no friction at joint between the two bones. Number two, shock absorbent and wound repair. Chondroitin acid, it is found in cartilage, bone and tendons, and its function is to provide shape and structure to cartilage, bone and tendons. Heparin, it is found in skin, lungs, intracellularly inside mast cells, blood, and its main function is to act as anticoagulant. Heparin sulfate, it is found in the Bowman's capsule of glomerulus, where it is responsible for the property of selective permeability of the wall of Bowman's capsule. One of the reasons why Bowman's capsule or the wall of the Bowman's capsule is selectively permeable is due to the presence of heparin sulfate. Keratin sulfate, it is found in cornea and cartilage. In cornea, it is due to keratin sulfate that it has the property of transparency, or the transparency of cornea is due to keratin sulfate. Similarly, it's also found in cartilage where it provides shape to the cartilage. And finally, there's dermatan sulfate, which is found in skin and heart valves. Dermatan sulfate provides shape and structure to your skin as well as heart valves. There are two terminologies that you need to know. One is proteoglycan and the other is glycoprotein. Both proteoglycan and glycoprotein contain protein plus carbohydrate. However, the difference lies in the composition. That is, proteoglycan contains majority carbohydrate. However, glycoprotein contains majority protein. Among these two, first let's deal with proteoglycan. So proteoglycan contains protein plus carbohydrate, where the portion of carbohydrate is more than 95%. So proteoglycan is majorly carbohydrate. But what does proteoglycan unit, a single proteoglycan monomer, look like? This is what it looks like. So under GAG, we saw that a GAG is composed of repeating disaccharide units, where one disaccharide unit contains two different sugar, one amino sugar and another uronic acid. Amino sugar plus uronic acid forms one disaccharide unit, and many disaccharide units together forms one polymer of many disaccharide units. So what I have represented here with the help of this green wiggly line is a polymer of disaccharide units, or a polymer of amino sugar plus uronic acid. Just like this, there are many other polymers of disaccharide units. And these many polymers of disaccharide units, they are attached covalently with a protein found at the core of the structure, so called core protein. So what's here? So here are the polymers of disaccharide units. That is, amino sugar plus uronic acid together is one disaccharide unit, and many disaccharide unit forms one polymer. There are many polymers, and these many polymers of disaccharide units are covalently attached to a protein that is present at the core of the structure, so called core protein. These two parts together, that is polymer of disaccharide units along with core protein, forms one proteoglycan monomer. And if you look at the structure, it kind of looks like a bottle brush. Do you know what a bottle brush is? So in a bottle brush, you can find a handle, and surrounding the middle portion or the middle metal rod of the handle are the feathers. So since it looks kind of like a bottle brush, we give it the structure of a bottle. We name the structure a bottle brush structure. So what you see here, the figure, is the bottle brush structure of proteoglycan monomer. So one proteoglycan monomer is actually composed of many polymers of disaccharide units, which is associated covalently to a core protein. So what you see right here is a monomer of proteoglycan. Many such monomers are found in an aggregate attached to hyaluronic acid. This middle rod-like structure that I have shown here using blue pen is the hyaluronic acid. Hyaluronic acid. To this hyaluronic acid, a number of proteoglycan monomers are attached. This right here is one proteolytic monomer. Proteolytic monomer. Multiple proteolytic monomer, this is one, this is another, this is another, this is another, are attached to hyaluronic acid to form an aggregate. Along with hyaluronic acid, there's your link protein, which is found at the base of proteolytic monomer. This link protein brings a link between the proteolytic monomer and the hyaluronic acid and makes the structure more stable. And you already know the structures present in proteoglycan monomer. There's your core protein and there's the polymer of disaccharide units. So polymer of disaccharide units. So this right here is proteoglycan aggregate. Similar to proteoglycan, there's another structure called glycoprotein. Both proteoglycan and glycoprotein is composed of carbohydrate and protein. However, in proteoglycan, carbohydrate is more than 95%, but in glycoprotein, protein part is more than 95%. In proteoglycan, carbohydrate is majority. In glycoprotein, protein part is majority. Glycoprotein is nothing but a protein which is found in association with or in covalent bond with oligosaccharide chain. So here's the oligosaccharide chain and here's the large protein. To the protein, oligosaccharide chain is found bound with the help of a covalent bond. Glycoprotein is found at numerous places in our body, such as enzymes, prothrombin, which helps in blood clotting, hormone, thyroid stimulating hormone, which stimulates the secretion of thyroxine from thyroid gland, antigen, antibodies, mucin, and collagen. And that's it for carbohydrates. See you again next time with a new video.